So when we look at humoral immunity and cell-mediated immunity, both of them rely on B cells and T cells. So with B cells, we have about 1 million different kinds of B cells, and T cells about 10 million different kinds. So when we look here at this B cell, I only have 7 or 8 of these Y-shaped antibodies on the surface, but in reality there's about 100,000 of them on each B cell. Here are some different ones. So each B cell, I have them shown here in different colors, but really the different colors represent the varieties of B cells I'd have. So here I have 3, but really we have 1 million different kinds of B cells. For the surface molecule here, this is called like an antigen receptor, but really these can be called antibodies. When they are released into our bloodstream, we call them antibodies. Oops, sorry. So here we have antibodies on the surface of our B cells, or antigen receptors. They're going to match perfectly with an antigen that's located on a pathogen. So during B cell development, to make 1 million different kinds of B cells, you would take the genes that code for these antibodies, because these antibodies here are really just proteins. So this is made of amino acids, it's proteins, which would then be coded for by your DNA, then makes mRNA, then makes a protein. So in order to get 1 million different varieties, since we only have about 30,000 genes, how do you get 1 million different kinds of antibodies? Is what we're going to do is we're going to rearrange the different genes. And so you get different kinds of patterns of the genes, which result in different mRNA and therefore different proteins. And therefore we have tons of variation in our antigen receptors. Okay, so now we're going to go into our humoral response. Now our humoral response is for pathogens that circulate or travel in our blood. We're going to use our B cells. The way I remember this is that B cells and blood both start with B. And we're going to use a process called clonal selection. So here I have my 3 B cells, but really there's about 1 million. And if I get exposed to an antigen, so here's an antigen on the surface of a pathogen, so something foreign inside my body, and as it's traveling through my body, either my blood or my lymphatic fluid. Let's say it's traveling through my lymphatic fluid and it passes by a lymph node, and in that lymph node I have some B cells hanging out. Now what will happen is that one of my 1 million B cells is going to be a match. So what will happen is this pathogen's antigens will match perfectly with the antibodies on the B cell. So this Y-shaped thing is going to be called an antibody when it's released. While it's attached to the B cell, we're calling it an antigen receptor. It's going to receive and attach to that antigen. The antigen here is red. So now the red triangle. So they match perfectly. And that's where in this name, clonal selection, where the selection comes from. So right here, the word selection, I've selected which B cell I need to defeat this microbe or this pathogen. So now once a B cell has been selected as a match, clones are going to be made, because I can't defeat a bacteria, virus, or other pathogen infecting me with just one B cell. So now I'm going to need to make a standing army. I'm going to need to make a whole bunch of clones of this B cell that can then go and help me fight off whatever pathogen is inside of me. So here's the process of how we do that. And this process is called clonal selection. So I've identified which B cell is the match and has antibodies that can defeat this pathogen. So now it's going to be signaled to start to divide by mitosis. How it gets signaled is at the end of the PowerPoint. Okay, so here it starts to go through mitosis and starts to multiply. Now these clones that are made is where this part of clonal selection comes into play. We begin to make clones of the identified B cell. This is like our standing army. This is what we're going to use to defeat that invader, that pathogen. Now these clones are going to divide into 2 groups. We're going to have one group that forms like our memory B cells, which would be like the Army Reserve, but maybe an Army Reserve made of veterans who've already seen the pathogen and know how to defeat it. And then we have our plasma cells. And our plasma cells are going to be like the ones that like hunt down our microbes, basically. Our plasma cells are going to do this by releasing antibodies. they're going to release the antibodies into our blood or into our lymphatic fluid, and those antibodies are going to attach to the microbe and help us get rid of it. So, as we see how that works, plasma cells are the ones that secrete antibodies that are specific for the antigen. Because remember, these antibodies here were selected for, they were a perfect match for whatever antigen is causing problems for us. So how this works is when the antibodies are released into our blood or lymphatic fluid from our plasma cells, they're going to attach on to the antigens on our pathogen. Now, this pathogen, if it was a bacteria, for example, it now cannot reproduce. It can't go through binary fission. It's been immobilized. Now, as we start to attach antibodies to antigens, we mark them for destruction, which would have a white blood cell come and pretty much destroy it and remove it and clean up the bacteria, or if it's a virus or any other microbe. So here's a quick little animation showing how the antibodies are going to help the pathogens to kind of aggregate and clump together, making it easier for a phagocyte to come like a macrophage and engulf them. So humoral immunity, I kind of like this because it shows us, like here's a Y-shaped antibody that would fit perfectly on this antigen, where this one is also specific for this type of antigen here. So there's specificity between antibodies and the antigen that they target. When we look at how this works, so here's a graph showing the first time we're exposed, where it says up here the initial exposure to an antigen. That means the primary or first time that we see a virus or a disease of some kind. So let's use like the chickenpox virus, for example. So my first exposure, I got the chickenpox, and then my body, like I felt I was sick for a while, and my body starts to build a defense, builds its army, and eventually defeats it. And so now I look on the left here, and it talks about antibody concentration. So over time, as my body builds its army, the antibodies increase and increase and increase as the plasma cells release them, and as you create more and more plasma cells. Now eventually I'm going to be fine. I defeat the virus, and now the levels of antibodies in my blood decrease simply because they're no longer needed. But remember, I did make also memory cells. They're going to have a memory of how to defeat this virus, this chickenpox. So, true story, 5th grade I got the chickenpox. 6th grade my best friend did. So if I'm hanging out with my best friend in 6th grade and she has the chickenpox, my second exposure, the second time my body sees this virus, my response is much faster. You can see it's a quick increase in my antibody concentration. Also, it's much faster. Look at the slope of the lines. It's a much faster, quicker response, and look how many antibodies I have inside my body at this time. So therefore, my memory cells are ready to go, and I defeat the pathogen much quicker, so fast actually that I don't actually get sick.